Alright, so today I'm going to be virtually showing you how you guys can complete your stream macroinvertebrate communities exercise. Uh, Please follow along and make sure to pay attention to everything I say, as it's very important in understanding how to complete your lab. So first things first, let's talk about due dates. Everything on Canvas should be updated. However, here's just recent due dates that you should keep in mind. If you can either pause the video and take a screenshot or just write it down. But basically, your invertebrate hypothesis of the lab for tomorrow, Monday the 14th, is going to be due tonight, 9 p.m. It should not be a surprise, as the professor emailed you guys that this is the lab that we're going to be doing. So the hypothesis is something that has to be done the night before. It's been this way since the start of the semester, so no surprise there. The one thing that has changed that's a little different is typically you take the quiz before the night before your lab. This time you're actually going to have a little more time. September 18th by 9 p.m. is when it's due. Your earthworms and soils lab report is still due the 18th by 6 p.m. You should have already known that from last week. And then the lab report that based off what we're going to be doing today is due the 21st by 6 p.m. And then on the 28th, we plan to resume face-to-face. However, that may change. I'll keep you guys updated. So I'm actually going to slide this over. So I'm going to go right into Canvas so that you guys know exactly where to find everything. So you just scroll down to the lab that we're doing, Stream Macroinvertebrate Communities. Now this is very important. There is 2 exercises listed. This is the one, the top one, is the one that was there originally. Now that we switched completely online, we're going to be going with the one that says simulation. So make sure not to go to this one. Go to the bottom, the second one, and use the one that says simulation. So we'll start a lesson. I expect you guys to really go through all of this. It's really important in understanding what this lab is about, as well as understanding the indices that you're going to be calculating. It's really important to go through each tab and understand that. Otherwise, you'll probably be pretty lost with the exercise. But we'll jump ahead because I already expect you guys to have already gone through and read through all that. So we'll go to hypothesis. So basically, we're just testing the macroinvertebrate taxa in unpolluted, moderately polluted, and severely polluted rivers in this simulation that you're going to be doing online. So for a hypothesis, all I'm looking for is in regards to species richness or the number of species, what is your educated guess on how that will change in unpolluted versus polluted? So guess, hypothesis, for example, would be when sampling unpolluted versus polluted rivers, the unpolluted rivers will have a higher number of species. I'm not saying that's correct or not correct. Just giving you an example of something along those lines of creating an educated guess on what you think the species richness is going to be when comparing unpolluted versus polluted rivers. So that should be pretty straightforward and easy. On to the actual objectives. So like I said, we're going to be doing a simulation. If you scroll down to this video, it will tell you how to run the simulation. I suggest going through it anyways, even though I will be showing you this. Maybe you can learn something from this video that you didn't already have. But if you actually go to here, we're going to be doing the simulation with the virtual biology lab project. The link to the actual simulation is here. If you go ahead and click it, it'll take you right to it. All right, so I'll blow this up once we start doing it, but for now, let me make this big. So lab objectives. So this is the screen that we're about to see when we start. This is how it should be default. Sampling time is going to be 500. You should probably start with the pollution at none. You can adjust the speed to see how fast, but I'll get into all of that in a second. The main thing is how you're actually going to be running the lab. So you're going to be doing each simulation a total of 30 times. you're going to do it 10 times for each pollution level. So 10 times for the none, 10 times for moderate, and 10 times for severe. Each sampling time, you'll keep it at the default set of 500. And what you're going to be doing is recording the total number of individuals of all these species into an Excel file. So I was really nice in that I spent time and I set up an Excel file which will calculate all your indices for you. But I'm still going to cover how I did that and what each one means because it's very important for you to actually understand it. The reason I decided to be nice and set up the Excel file is because I know not everyone is used to Excel, and switching to an online portion, I can't really gauge where you guys are at. And I know if I would have just left it for you guys to do by yourself, I would have got a lot of questions. So it's going to be pretty straightforward where you're just going to be plugging in numbers and taking numbers that are already calculated for you. However, for your lab report, you're going to need to understand what those numbers mean and exactly how you got them. So we're going to be calculating species richness, relative abundance, Simpson's index, Shannon-Weiner index, and a macroinvertebrate biotic index, or MBI. So I'm going to exit out of here real quick. And we'll actually go into the simulation itself. So this is the screen that you have. Let's say we're going to start our very first one. You want to set the pollution to what you want it at. I suggest probably starting with none, doing 10 of those, and then 10 of moderate, and then 10 of severe, just because it goes like that in order to keep it simple. So make sure that your sampling time is at 500. You want to click go, and this will start having the invertebrates flow down. Now, if you notice, nothing is actually changing down here. That's because you need to open your seine. If you open your seine, you'll start collecting them. And so you have to keep doing this until your sampling time is 500, and then it'll stop. You can open and close your seine. If you close your seine, it'll just pause. But you have to have it open for a total of 500 seconds for one sample. So you can actually speed this up a bit. The faster you go, it might lag your computer a little bit, but for the sake of the video, I'm going to go pretty fast to try to get it to end. So it does get a little choppy, but you'll see that it pretty much continues to work. And now it's at 500 and it stopped. Your seine's gone. So just so that you're not lagging as much, I would lower it again. So this is kind of a bad example, but one thing that you need to know is that gill snail, which of course we didn't catch any in this one, normally if you do get gill snails, there's an error with the system. It doesn't calculate it for you, the number. So you're going to have to make sure to actually count these. We'll go ahead and do another one later on, and maybe we'll get some, and I can show you what I mean. But like for an example for here, there's obviously 3 of these in here, and it correlates to the boxes here. So the boxes up here are correlated to here. So this is why this one says 0, because there's none in there. But typically, even if you get some, it'll still say 0. It's like a glitch in the system. So make sure to count those for your numbers. So you just completed one sample. So you have your 500 sampling time. You caught a total of 15 species and a catch of 86. So what I'm going to do now is here is the Excel file that I created for you guys. So everything's laid out. All you have to do is plug in the numbers for the samples, for the 10 samples for each type. So this is for none, and then obviously if you go down here and go down here, it switches. It's all the same file, but you need to make sure that you get 10 samples for each. So we just did our first sample. So we can go over here, and all we're doing is getting the number that it says. And it should go, the way I set up these is how they kind of go in order. So you can just go right here. So caddisfly, I had 10. Mayfly, I had 4. Stonefly 9, 1, 3, 0 gill snails, so we can leave that blank. Dobson fly 6, crane fly 4, 5, 7, 5, 3, 2, 5, 5, and 17. So you just want to make sure that you double check, make sure all your numbers line up. A way to quickly know is you can highlight that, and down here I'll say a sum of 86 and a count of 15. We had 15, 86, so it checks out. So as you can see, this already did a bunch of calculations. Now, I'm going to pull up in here the actual calculations and break it down for you. All right, so first off, we're going to do relative abundance. So relative abundance is explained right around here. I don't want you guys to get intimidated by any of these advanced looking equations. They're actually not bad at all. Basically, I broke down in my actual presentation what each one means. I'm going to send this in the email as well, so you can go back and look. But basically, relative abundance is PI, and all it's doing is taking the number of individual species divided by the total number of individuals. So that's all this is saying. Ni being the number of individual species, and you're going to divide it by the sum of the total number of individuals. So if we go back to our Excel sheet, PI was calculated by taking the total caddisfly, 10, divided by the grand total, 86. So when you're actually doing it, make sure not to touch any of this stuff because the formulas are already in there. If you click anywhere, it'll mess it up. So if you do happen to click it by accident, don't click anywhere, just click enter, and it'll be fine. So that's how right here, this is how we calculated relative abundance for everything. So I'm showing you just an example for just one sample. What you're going to be doing is running that same, hold on, I'll go to it. Running this 9 more times and collecting the data for each sample and filling all this in before you actually look at your calculations. But I obviously am not going to sit here and do 10 or a total of 30 because 10, 10, 10. For the sake of time, I'm just going to do the one sample and show you. So that is basically what we're doing for relative abundance. So species richness, this is just going to take account if there's a number. This column really doesn't really matter. It's just kind of to get you guys understanding that species richness, I wrote down here, is just the number of species. So this is actually wrong. I may have to take this out because what it's going to do is calculate whenever there's a number here, and you're really not going to get, let's say you have 10 samples and you have a number here, a number here. It's going to say that there's 3 species. That's actually wrong. So for yours, species richness is not going to be in here. I'll get rid of that because that's actually confusing. But basically, it's just the number of species. So you can look here, for our first sample, there's a total of 16 different ones, and we had 15. So our species richness was 15. We had 15 number of species, if that makes more sense. So then over here is where we get into some of the other calculations. So the Simpson's index. Once again, it's going to be under the species diversity indices. It is right here. Once more, don't get intimidated by the formula. It's actually very easy. All it's taking is the relative abundance, which we already calculated, which is PI. So this was the relative abundance. It's taking PI squared and adding all of them together for all of them. And then you're just doing 1 minus that. So it's 1 minus the sum of relative abundance squared. So I made a column here that calculates PI squared. So this is PI squared for all of them. And so all this formula is doing is taking the sum of all of them, because they're already squared, so we already did that step. Now it's just adding all of them, and it's going 1 minus the sum of PI squared. And that gives you 0.904. And that's how you get your Simpson's index. It is on a range from 0 to 1. 0 means all the individuals are the same species, and 1 equals all individuals are different species. So if you want to just mentally check to see if that makes sense, well, we got 15 different species out of the 16 we possibly could have gotten, and there's a good amount of variation, so it makes sense that we would get a close to 1, because it is close to having almost all different species. If you scroll down to the Shannon-Weiner index, this one is a little tougher. So it's going to take relative abundance times log E, which is then going to be of PI again. And you're going to take the negative sum of all that. So I broke it down into steps. Log E is just going to be ln times PI. So in this case, it's just ln times PI. So we got all of those. Then to get PI, so relative abundance times that, I just took that times PI, and we got that step done. So that has all of them. And so this is just the sum of all of that with a negative in the front. Now, if you notice, right now it says number. It's not actually giving us the appropriate calculation. That's because it's factoring in a 0 for this, which we didn't get any species. And if you go into here, their example where they're doing it, they just take it. This is their example. There's obviously a long list of species that they could have gotten. They didn't put 0 for all the ones that they didn't get. They just used the numbers of the species they actually got. And so if you go down to their calculation, they used the species of that they got using the relative abundance number. So ours is actually calculating 0. Once you have all of your information in, if you have any that are a 0, you need to just remove it. So just highlight it and delete it. And then you see now it suddenly gives us an actual number. Because like I said, you only want to calculate for the species that you actually got. A 0 is going to throw it off because you're saying that you didn't get the species and you're counting it as if you got one, but you actually didn't. It's not really 0. It's just a species you didn't get. So once you have all samples, all the 10 samples done, if you have any that the total comes out to 0, you're going to want to get rid of that whole row of stuff because it's going to affect the calculation here. But as you can see, we removed it, and now it gave us 2.517. And so the way Shannon-Weiner index, it actually doesn't have a scale. 0 is the lowest, and then it has a limited of how high it can go. The higher the number, the higher the diversity. So based off just this number right here, we can't say much. what we can do is when we do the same thing for moderate and the same thing for severe, we can compare the number that we get for that, and whatever is the highest means that it has a higher number in diversity. That's all that's saying. And so now we can move on to our MBI, Macroinvertebrate Biotic Index. So for this calculation, when you go into Canvas, you're going to want to, as its own tab, explaining all of it. Once again, it's very easy. All it's doing is grouping the actual invertebrates into groups based on how sensitive they are to pollution. The more sensitive they are to pollution, the higher rating they get. So if you look in the calculation, A is going to be the total amount of organisms in a sample that were pollution sensitive. So because they're sensitive, if you have them, they're going to have a higher rating because that you want it to affect your rating more because it's showing that the water quality is good if they're present. If they're sensitive to pollution and they're present, it means that the water doesn't have pollution in it. So they're going to get a higher rating. So A in the formula will get times 3. B is going to be pollution less sensitive. So they're more adaptable to water that has pollution in it. So they're only going to get a rating of 2. And then C, which is pollution tolerant, they can exist in bad water. They're only going to get a rating of 1. So it does say you can go to, and it'll tell you in here to refer to the appendix to see what species are in which group, which I'll go ahead and do. And if you scroll down, right here it tells you what group they're in. I have this picture here. This is basically just this. And so all the first ones are pollution sensitive species, so they're in group A. All these are group B, and all these are group C. So going back to the formula, you're just going to get the total sum of all of these, and that will be what A is. So let's go to Excel. Once again, I already did this for you. So all it's doing is taking the sum of all these. So I made red as the pollution sensitive species. All of these in the orange yellow is pollution less sensitive species, and then the green are our tolerant species. So if you look here, A is taking all of these, just the sum of all of them. B is taking the sum of all of those, and C is taking the sum of all of those. Now, going to the formula, it's going to be A times 3 plus B times 2 plus C times 1, and you're going to divide that by A plus B plus C. So if you go to MBI right here, I did the formula for you. So it's A, it's taking that times 3, N20 times 2. Behind this would be N21 times 1 divided by A plus B plus C. And it gives you a rating. And so for this, it's 1.9. And so once again, just to clarify, you're not going to be doing any of this and looking at any of these numbers until you have all 10 samples in. Once you have all 10 samples in, then you're going to be looking at the numbers. But once again, I'm just showing an example using just one. So all of this was calculated already for you, and it's right here is the formula MBI. So we got a rating of 1.9. So what does that mean? If you go down to here, when we're in this tab, the MBI tab, it will tell you what each one means. So greater than 1.75 are considered to have poor water quality. Sites with MBIs of 1.76 to 2.25 are intermediate sites, and sites that are greater than 2.25 are good sites. So we got a 1.9, which means we fall into the middle category, meaning that this is an intermediate site. So that is how you do, how all these calculations were formed. Down here I wrote kind of examples of what each of these mean. So like PI is relative abundance, species richness is just the number of species, D is the Simpson's index, the H with this is Shannon-Weiner index, MBI is your macroinvertebrate biotic index, and then I explained ABC, what each one means again, which it just correlates to this table, and I already separated it for you. So everything is done for you. All you have to do is just run your 30 different samples. So you'll go in and do 9 more, fill all that out. Then you'll go to moderate, which means when you go back to this, you're going to want to change this to moderate. Make sure that it's sampling times 500. All you got to do is make sure to do reset. Once you reset, all your numbers are gone. And you just, once again, click go. And so you can speed this up and open your Sane, click go, and you're ready to go. Notice how mine did kind of lag there for a second. It's because I had my speed all the way up. Just to show an example. So once again, you have all your numbers. You're going to want to go in underneath the moderate tab, and you would just do the same thing for sample 1, and you would redo this 9 more times to get your full 10 samples. Fill it all out, and then you'll actually look at the numbers. Keeping in mind that if you have any zeros within here for your total, you're going to want to remove that column because you don't want it incorporating a 0 in your formula, or else it'll mess it up and you won't get the appropriate measurement. Not sure why that just opened. Oh. So going back to what I was talking about, the gill snail, just another reminder. This time we actually got them. So they're in here. But notice how it says 0. You need to make sure that you don't mess up and put 0 when you're copying in your information into your Excel sheet. So you need to make sure to count. So as you can see, there's actually 3. It's just an error in here that it says it's 0, but there's really not. Just make sure when you're putting in for gill snail that you put 3. That's pretty much exactly what you guys are going to be running through. You can go back to here and go to your lab report. You're going to be keeping these questions in mind when answering. Did the data support your hypothesis? How did an increase in pollution level affect the composition and diversity of the macroinvertebrate communities? How different in the composition were the polluted streams from the non-polluted streams? Which species benefited from pollution and which were adversely affected by pollution? How well did the MBIs reflect the level of pollution? So you should address all these within your discussion. Don't answer it 1, 2, 3, 4. Answer it as a paragraph format. Make sure to talk about your data. Don't just answer just this. I want your discussion to still include explaining your results, because your results section is just listing the results. Something to keep in mind is that you need to make sure that you're doing a full lab report. You should go to writing lab reports, go to the exercise, and refer to this for information on how to set everything up. So I should see everything set up exactly how it's describing to for all of these sections. So you should have a title and author, an abstract, an introduction, study site, methods, if it's applicable, results, discussion. Literature cited won't be needed for this. If you're wondering about the grading rubric, it's listed here, and it says exactly what you need for each to get 100. There's an example report here that I would refer to. it's really, it may seem like a lot, but it's not that hard. You just need to follow along the steps here and you'll do fine. Uh, but if you don't include certain things within it, just looking at the grading rubric, let's say you just forget your abstract, that's already 10 points off. That's an easy 10 points. Make sure to include everything. Make sure to actually label it as so. Um, I want to not guess whether that's your abstract or not. Just put abstract. So, all right, this is where their abstract is. This is where their introduction is. Keep it really simple. Um, one thing to keep in mind is you should have known from the email, but just to reiterate, it doesn't matter what group you're in. For the next week, next 2 weeks, you're going to be, whether you're in gold or cardinal group, you're going to have to do the lab for each week. So this week is the stream of macroinvertebrates. Next week's going to be a whole nother lab. So gold group isn't waiting a week to do this lab. You guys have to do this lab this week. It doesn't matter what group you're in. I know gold typically wouldn't have met this week, but because we're online, it doesn't matter. I want to address that September 28th, we plan to go back to face-to-face. I will keep you updated on how that plans out. Things can change. And so if anything does change, I will let you guys know. But as of right now, we're only going to be online for 2 weeks. If you have any questions at all, please feel free to email me. I know it's kind of a lot to take in, but I think I really helped you guys out a lot by setting up this Excel sheet. It pretty much does everything for you. All you have to do is fill in the samples, the 10 samples for each of these, and keep in mind the two things that I mentioned, that with the gill snail, you need to manually count them. And if you have any zeros after the 10 samples, you got to get rid of it to properly get the right calculation. So yeah, if you have any questions at all, please email me. I will help you and answer you right away. Monday, I will be available during the typical lab hours, so I will probably get back to you fairly quick if you answer during your normal lab section. If you answer outside, I'll try to get back to you as soon as I can. But I think this, me recording myself will help you guys a lot, and that if you get confused for anything, you can just go back and rewatch the video. You can also have all the videos on Canvas that you can refer to on how to actually do it, such as how to run this. So you should be in good hands. Uh, but if you have any questions, let me know. But make sure to read through all of this, as I shown going through everything, that it is very helpful in understanding how these indices work and how to actually properly do the lab. But I guess that's pretty much all I have. So hopefully you guys understood everything. And if not, send me an email. And that's about it. Thanks, guys.